Hello everyone, welcome back to the channel Xgene and proteinX. In this tutorial video, I'm going to talk about the overview of the DNA, RNA, chromosome, gene, transcription and translation. So let's begin. So first, what is DNA? So DNA actually is the deoxyribonucleic acid and is a polymer composed of two polynucleotide chains that coil around each other to form a double helix structure, and it carries the genetic instruction for the development, function, growth, and reproduction of all known organisms. So if we analyze this term deoxyribonucleic acid, so first we can divide it deoxy, so I'll explain it later, and then is a ribo. So ribo is for the DNA backbone contain the ribose sugar, and the structure of the ribose sugar is this 5-membered ring. And if we look at this double helix structure, then you can understand that this backbone is composed of this ribose sugar along with the phosphate group. And next is the nucleic acid. So nucleic acid or nucleotide, there are 4 different nitrogen bases nucleotide present. These are the adenine, thymine, guanine, and cytosine. So you see in this figure in between, and this adenine, thymine, and guanine, cytosine paired with hydrogen bond. So next I will talk about the what is a deoxy. So deoxy actually, so ribose here in the number 2 position there is one oxygen group present, so OH group, but in the deoxy it means oxygen is reduced from there, so that is known as the deoxyribose. That's what. So this deoxyribose sugar is present in DNA, not the ribose sugar, but this ribose one that is present in the RNA. So this is why deoxyribose is present in the DNA. This is why it's known as the deoxy. And for ribose sugar, that is for the ribo, and for the nucleotide bases, that is known as, that is for nucleic acid. And next is how exactly nucleotide bases are paired. So as I mentioned earlier, there are 4 different bases: pyrimidine and pyrimidine, thymine and cytosine are pyrimidine bases, and guanine and adenine purine bases. And also there are one ribose sugar along with the phosphate group. So this DNA contain this nitrogen bases along with the ribose sugar and phosphate group. So let's look at how these bases paired with each other. So you see here the adenine and thymine, these are paired with two hydrogen bond, and cytosine and guanine paired with 3 hydrogen bond, and the backbone, these are composed of ribose sugar along with the phosphate group on both sides. So next, then if you look very carefully, then you can understand that this backbone is for sugar base, for this backbone for ribose sugar and phosphate, and in between these bases that paired with hydrogen bonds, 3 and 2 hydrogen bonds. So next is what is chromosome. So chromosome actually is a structure found inside the nucleus of a cell, and a chromosome actually is made of proteins and actually is organized DNA, very well organized manner, and each human in each cell normally contains 23 pairs of the chromosomes in human. So if we look here at how this chromosome present in the nucleus, so if you zoom in, then if you zoom in here, then how this chromosome looks like. So this chromosome, each single chromosome is called chromatid, and this chromosome, this black portion is called loci. So these loci are the particular genes are present in the loci. So basically the chromosome, main function of the chromosome, in the DNA is very organized by the chromosome. So this is the 23 pairs of the chromosome that found in the human, including X and Y chromosome. And next is what is loci that I mentioned on my previous slide. So loci is the plural form and locus is the specific and fixed position of the chromosome where particular gene is located. So if you look at this figure, that this is the one of the chromatid, and here this is the P arm and Q arm, and if we divide it in particular segment, then it is known as a P3 3p22 because this is a P arm and this is number chromosome 3, that's what is 3. And if we further segment it, then it is known as the 3p22.1. So here as you see, there are a lot of, in this chromosome contain a lot of loci. So this loci also is for genes are located in the loci. And then what is gene? Gene, actually this is the long fragment of the DNA, and that is the segment, a particular segment are called gene. So this is the long fragment of the DNA, and it contains some specific fragment. These are known as the gene, and this specific sequence of the DNA that role for particular gene and that code for particular protein. So here in this loci contain lot of genes present. And then human DNA consists of about 3 billion bases, length is 1.8 meter, approx 5 feet, and the nucleus is around 10 micrometer diameter, very tiny. Then question is that how can a very long DNA molecules fit into a tiny nucleus? So if you look at this figure, that this chromosome is the DNA sequence organized by the chromosome, and this DNA sequence, a very long DNA sequence, is wrapped by the histone protein. So this is why the concept of the histone proteins comes here. So histone, around the histone proteins, this DNA sequence wrapped tightly. So this is why it's possible to fit this long DNA sequence in the tiny nucleus. So if you look at here, that this nucleus, if you zoom in, then this contain this nucleolus, and this nucleolus contain the genetic material, our chromosome. And this is the long strand, long sequence of the DNA, and then this DNA actually is wrapped by the histone protein. So if you look here, so how exactly these histone proteins and these genes are wrapped around these histone proteins, and these small fragments are called the gene. But when these genes are, when this DNA sequence are wrapped tightly, then that DNA sequence cannot express protein. So this is why, so this part, if you see here, so this part, so this red figure, this is wrapped, so it cannot access, so this is inactive form. So this histone can, it's by unwrapping this DNA sequence by acetylation or deacetylation, that is known as the epigenetic regulation, and by this epigenetic regulation, gene is wrapped or unwrapped, and then during the expression of the protein, then the particular DNA sequence is unwrapped by the histone protein. Then what is RNA? RNA actually is the ribonucleic acid. It's a polymeric molecule essential in various biological roles for coding, decoding, regulation, and expression of the genes. And if we analyze this term ribonucleic acid, then ribo is the ribose sugar, and ribose sugar, and here is the oxygen group in the ribose sugar, but in the there is oxygen is not present in the deoxyribose. This is why the main comparison in between RNA and DNA. And the nucleic acid, so the nucleotides that present in the for the RNA, but for RNA that is a little different from the DNA, because in RNA contain 4 nucleotides that are but thymine is not present. So instead of thymine, in RNA there is uracil present. So main comparison are here, that then DNA is double stranded, RNA is single stranded, and DNA contain thymine but RNA contain uracil. DNA contain there is one this DNA contain this ribose sugar deoxy, but here is the ribose sugar, the oxygen present. So let's have a look at the kinds of types of the RNA: messenger RNA, transport RNA, and ribosomal RNA. So how do genes direct the production of the proteins? So there are two different pathways: transcription and translation. So transcription is a process, and transcription and translation, this together is known as the gene expression. So during the process of transcription, the information stored in gene's DNA is passed to a similar molecule that is called the messenger RNA, and that is copied during the transcription. So as you see in this figure, this mRNA is copied from the DNA, and then this transcription is carried out in the nucleus, but this mRNA, it moves to the endoplasmic reticulum for protein synthesis. So here, as you see, the DNA contains these nucleotide bases, and during transcription, these specific codes are copied from the DNA, and that forms the messenger RNA. And these three nucleotide bases are called codon. So codon is a sequence of 3 DNA or RNA nucleotide that corresponds with a specific amino acid or stop signal during protein synthesis. And then the next process is the translation. So these specific codons that code for specific amino acid. So this translation is actually the process from the mRNA to the protein synthesis, and this process is carried out in the endoplasmic reticulum. The ribosome, it can detect these codons and then synthesize the proteins. So, but for every protein there is one start codon and one stop codon. So this is around 64 in total codons present, and that codons code for 20 amino acid. So these three are codons for stop codon, and one is for start codon. So next, you have a question. You might have a question that what is the percent of the genome identity in between human and monkey, and individual of the humans, and human and gorilla. In the monkey and human, the percentage of the identity is 98.8%. That is very big similarity. And the gorilla and human, there is 98% similarity. And in between humans, individual of the humans, there are 99.5% similar. So only for 0.5% sequence difference are used to distinguish all humans. So this is all about this overview. So I hope this video will be helpful. So if you find so, kindly hit the like button, share it, and subscribe my channel. Thanks.